Here we have chapter 11. This looks at how information is passed on from one another. Okay, we know mitosis, we know meiosis, we know the steps. This is going to lead us into that central dogma of biology of how we get from DNA to the end result protein. Because that's all DNA is. DNA is just information for proteins. Proteins code for structures. Proteins do things throughout the entire body. Misread a protein, you potentially don't have normal function in the cells and the person can't survive. Okay. So it's all about understanding how this information is passed on from one generation to the next. That's where Gregor Mendel comes in. Gregor Mendel it's pretty interesting, his story. He wanted to go to higher education schools to become a scientist. And he never had any real formal training in science classes. And so he was never really allowed entry into some kind of post-secondary education. So he became a mathematics individual. He was a math dude. Well, he was also a monk, and you can imagine had a lot of free time on his hands. And he loved to garden, and he did a bunch of kind of studies and research into how information was passed from one generation to the next in plants. At the time, it was this concept of blending, okay, where parents produced offspring and it was just a blending of the two pieces of information, half from mom, half from dad, blend it together, ta-da, that gets you. His research, which I don't believe was really embraced at the time, because no one really understood some of this actual significance of his stuff, and it really didn't become much more accepted until like the 1900s. But he formulated what was called a particulate theory. There were some particles. He didn't really understand what the hell those things were, and these particles were actually passing these inherited traits. Okay, so he came up with a couple of different laws, and sometimes these laws and describing them makes a good short answer question. Sometimes they don't. It just depends on the semester. If you can do it though, you can answer any question. Law of segregation, law of independent assortment. Okay, it's just how the genes reshuffle. Okay, one, they line up at the metaphase plate. Well, it's not that this one always goes here and this one always goes here, and then this one always separates this way. It's random. Okay, so we've kind of seen these concepts, but now he's putting some names to it, and that's all it is. Okay, all of that lining up at the metaphase plate happens by happenstance. Okay, they line up independently, they separate independently. Okay, so that's where these two laws come from. So here's what, I just love it because it's a picture of a monk, Mr. Gregor Mendel. So that's why I have it in there. But he did a lot of work with peas, Pisa sativio. Okay, peas are easy. I think I have it on the next slide, just why they're just easily manipulated. There you can kind of see Austrian monk, and he was very meticulous, ton of data, whatever you want to call it. And you could see when he did his stuff in the early 1860s is kind of when he put this all together. And, evolution didn't really become a thing until the 1900s. So he didn't have a microscope. He didn't understand any of these concepts of what were actually being passed on from one generation to the next. And later on in this chapter here, we'll see not everything was as easy as this. Not every single trait in us, animals, is as straightforward as what he thought it was. Well, we'll talk about it later, but there's a lot of genes that, multiple genes can code for stuff, and there's codominance, and there's whole bunches of stuff that he did not even know, didn't even understand, to be honest. So why did he use peas? Easy to grow, easy to manipulate, very short generation. So you want to have a short generation so you can get a lot of data points. They're normally self-pollinating, but he can cross-pollinate by hand. He can apply the pollen specifically from one plant to another and look for very specific outcomes. So in that regard, they're very easy to manipulate. All right. True breeding. Okay. They're true breeders. Okay. Purple gives me purple. White always gives me white. Okay. That's what we mean by true breeders. Tall is always tall. Short is always short. Okay. Very simple, very objectable traits that you can easily see. And there's not, well, it's not as tall as this. It's maybe in between. So it's a medium. No, it's either tall or short. It's either purple or white. Okay. Very easy, very simple. That's why he used it. And here you can kind of see, you can cut the anthers away and then you can brush the pollen on there and you can get very specific outcomes. Okay. I ain't going to ask you anything about this. I just thought it was interesting. Okay. At least this picture. Okay. I'm not going to ask, how do you cross pollinate a pea? I ain't going to do that. So let's look at his laws. Okay. To get this first law of segregation, okay, he used a one trait cross. Okay, mono means one. Okay, he used a one trait cross. True breeders. Tall, short, easy. And that's what I show right here. Okay, tall, short. Okay, they were homozygous. And I should probably have put what these words mean first, so it'll make a little bit more sense. But that's not how the book has it set up, and I apologize. Okay. These are plants that came from plants that were tall and produced nothing but tall. Homozygous. Okay. I can't see where they're at. Okay. Homo means same. Okay. Homo means same. So these always produced tall plants, no matter what. There's some shorthand that they use. The parental generation is always represented as a P. Okay. The first filial generation, the first offspring generation, okay, is called F1, the first generation. And then the second generation is always represented as F2. Okay. On the exam, I may have some Punnett squares up. I may have some P's, some F1s, some F2s labeled. So you have to know kind of what these are, because I'll ask, what's the phenotype and genotype of the P generation, or the F1, or the F2, or the F3, or yada, yada, yada. Okay. So you will have to know what those shorthands means. Okay. So to come up with this law of segregation, he used this one trait cross. Okay. And here we'll kind of look at it here. Sorry, there we go. Homozygous. homozygous. Okay, they are the same, all tall. So he used two letters, an uppercase and a lowercase, well, I guess one letter, two different forms, to represent tall and short. Okay, homozygous, homozygous. Okay, homozygous tall, homozygous short. Cross those two true breeders. In his in his first offspring, the first generation that was formed, everybody was tall. Everybody was tall. Okay. If I put that in a Punnett square, this is what it would look like, right? Big T, little t, the tall and the short, okay? And then if I just fill in my Punnett square, right? Pretty easy. Everybody is tall. But what are they? They're heterozygous. Okay. Everybody's still tall. So what should that immediately tell you? Big T, and big T, little t are both tall. Okay. Little t is the only one that's short. Okay. So this is very easy, very straightforward. Then he took two individuals from this first generation, crossed them. Okay. Again, big T, little t, crossed them, tall, tall, tall. But here's that little runt, a short one. So there's always a 3-to-1 ratio with this monohybrid type of cross for this one trait that we're looking at. We're only looking at one trait, tall, short, okay? We're not looking at colors. We're not looking at peas in a pod. We are just looking at one trait. Very straightforward, very easy to see. They're either all tall or they're all short. How much is it? So we understand this can only happen if this recessive allele is masked in that first generation. It basically doesn't code for anything. It is a recessive trait. Okay. And this is a dominant trait. Okay. Dominant trait. Dominant always is going to dominate over the recessive allele, the recessive trait. So here in that second generation, he got a small little dude. That's where he was like, okay, these traits from the homozygous plant here, from that parental generation, segregated, okay? But when they came together, they all showed tall. So these are true breeders. They were either going to be tall, even if they were heterozygous or not. and we still keep this recessive allele in the population, which is a good thing. Okay, gives us variation, gives us adaptability, and that's key for evolution. So what is the law of segregation? Each individual has a pair of factors. He used factors. We use the word alleles now, okay? Okay. And those would be those letters that we were utilizing. Okay. These factors segregate, or these alleles separate during the formation of the egg and the sperm. We saw that with meiosis. Okay. Each gamete has half the material, right? Half from mom, half from dad. So mom has an allele. Dad has an allele. The alleles come together and ba-bam, get you. This is what basically disproved that blended theory. Okay. If it was a blended theory, okay, they would have all been something in between. All right. But they were all tall. They weren't medium. Okay. They were all tall. So this law of segregation, this monohybrid cross, this one trait cross, is what basically disproved that blended inheritance theory. Okay. The vast majority of traits when it comes to the P are controlled by just two of those alleles. Okay. One was dominant, capital, and one is recessive, lowercase. Okay. But when it comes to traits, they all need at least two alleles. Those can be homozygous, homozygous dominant, homozygous recessive, HD, HR. Okay. But they are identical. Or they could be hetero. Okay. Two different. One's tall, one's small. And this is what we have mapped out. Okay. Human Genome Project, that's what mapped out the entire genome. So we know the specific allele for every chromosome. Okay. We may not know exactly what all of them do 100%, but we know where everything codes for and the processes from which those alleles give us information for. And we know it for a number of other organisms as well. Okay. Various different plants and animals. And this just gives you some stats. You can probably just block this out of your mind. We have dominant, we have recessive alleles. And alleles are genes that code for specific traits. There's also something you're going to be asked to probably tell me on an exam. I'm probably going to ask you some potential genotypes and potential phenotypes. What the hell are those things? Well, genotype refers to the genes. Okay. What are the alleles? Okay. What are the genes? What are the genes? Okay. Genotype. I can ask for, what do we see? What's the physical P, okay? What is the phenotype? What is the physical appearance? What do you see? Okay, what do you see? Do you see tall? Do you see yellow? Purple? Blue eyes, brown eyes, black hair, dark hair, blonde hair? Okay, what do you see? So for all of these crosses, there is a genotype. Okay. Or what do we see? What are the characteristics? Tall, short, round, wrinkled, yellow, green, purple, white, yada, 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 yada. Okay. What is the genotype? What are the alleles? Okay. Or what do we see? What is the trait that the individual is showing? We know what the hell all this means now. Gregor Mendel had no idea. Okay. He didn't know about DNA. Okay. What we do know now, okay, because he was using, he was using particles and factors, and he didn't understand what was actually passing this information from one generation to the next. Okay. But now we know what those factors are. Those factors are alleles. Those alleles represent the DNA, the genetic material that gives us the specific words to put together a protein, to get me something tall, or to get me something purple, or white, or green, or yellow, or whatever it is the trait codes for. The dominant allele always prevails, okay? The dominant allele always prevails. The dominant allele will always show the trait, okay? The dominant allele always does the coding, all right? The recessive allele, it represents basically a loss of function, okay? We carry the trait, but the trait does not manifest in somebody, okay? That's what that means. Color blindness is a great example of that, okay? If I don't have the dominant allele for normal color vision, okay, if I'm missing that, and the only one that's passed on is a recessive allele, you're colorblind, all right? Since there's no dominant allele to mask it, it will be presented, okay? And this kind of helps us to really understand meiosis and its true function into helping to pass information on to the next offspring. The last law we have here, guys, and we'll stop after this, okay, is this law of independent assortment. With this one, Mendel had to go and basically look at two different traits, okay? So he used what was called a dihybrid cross. Di means two, okay? So this is a two-trait cross, okay? This is a two-trait cross. So he looked at individuals who were tall and short, and either the pea pod color or the flower that it produced color, okay? And he did the same thing. Took two true breeders, okay, that always showed that in the line, and I can't remember specifically what he showed, what he did. Yep, okay, tall and short, green pod and yellow pod, okay? So he took two true breeders, just like he did in the monohybrid cross. Did the same thing here, okay? Did the whole self-pollinating thing, observed things, crossed the F2 generation to see this law of independent assortment. Okay. Whoops, sorry. What in the world did Mendel mean by these laws of independent assortment? Okay. These factors segregate independently of each other and of the factors. Okay. So if I kind of spell it out here for you, okay, we need 1 and 1. Okay. I need one allele for each of these traits to be included in the gametes. So it could be this guy and this guy. It could be this guy and this guy. It could be this guy and this guy. Or this guy and this guy. They independently assort. But these are true breeders. So what's it always going to be? It's always going to be that. Okay. 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 Same thing over here. Little t, little t, little g, true breeders, right? Here and here, here and here, here and here, here and here. And then you just plug and chug, right? But what in the world are these all going to be? These are all going to be big T, big G, little g. Okay. And what did we get? Oh my God, the same thing. The dominant allele won out, just like it did in the monohybrid cross. Okay. Took 2 from this first generation, crossed them. What did he get? Holy smokes, a 9-to-3 to 3-to-1 ratio. A disaster. Tall green. Tall green. Tall green. Tall green. Tall green. Tall green. Are we seeing it? It's the same dang thing. Okay. Tall green. Tall green, right? The dominant allele wins. All right. The dominant allele wins. But these 2 factors code for 2 separate different things. And they separate independently. Okay. This one may go with this one. Or now here, right? Big T, big G, little g. Could have a big T, big G. Okay. Big T. Could have a big T, little g. Oh, shoot. Look at that. Independent assortment. Could have a little t, big G. Could have a little t, little g. Independent assortment. Okay. These things separated independently from one another. All right. What does that look like if we're talking about meiosis and lining up? Yeah. Okay. They can line up anywhere. Okay. Independently separate, independently assort. Okay. And that gives us the varied difference. And again, what do? We just put those into a Punnett square, right? We put those into a Punnett square, and then we can figure this all out. That's what all of these are here. This is a 16 square Punnett square. Okay. If you're doing a 2-trait cross, it's always going to be a 16 square. All right. I know that's probably a lot. Okay. Take it, read it, rewatch the videos. Okay.